Good afternoon. I'm Masayaki Fukumura, CEO of Biocomo. I would like to appreciate inviting me the presentation. And by using a platform virus vector, vector named BCPF, derived from the human respiratory parainfluenza virus 2. We have developed the vaccine for infection and immune oncology. Nowadays, at first, I would like to give the property of the BCP vectors. Secondly, the RSV vaccine, and then immune oncology reagent. BCP features a replication-deficient RNA vector that can benefit delivering the ectopic large protein as well as ectopic gene. Other properties is like this. Regarding vector system, we are often asked what is the strength of BCP compared to the messenger RNA vector. BCP is especially characterized by the nasal administration and can strongly induce a mucosal and secretory IgA in the upper respiratory tract in addition to the serum IgG. I will show you the example of the intranasal corona vaccine by BCP. Like this, COVID-19 infection can strongly protect the nasal turbinates at the virus entry site, as well as lung by intranasal administration of the BCP with a spike protein of COVID-19. And I'd like to move on to talk about the RSV vaccine. RSV is an infection that occasionally causes serious symptoms in infants and elderly. Therefore, licensed RSV vaccines are required for a long time. For the elderly, the RSV, Abrysvo, and other vaccines were licensed recently. And the sales of RSV led to blockbusters. However, many attempts to develop the vaccine for infants are about to continue to the failure. The person concerned tell us that you should give up the vaccine development. Lately, that messenger RNA vaccine for infants was interrupted. On the positive note, it was reported that a nasal vaccine for infants using the parainfluenza virus 5 was successful in the phase 2 clinical trial. We estimated mucosal immunity by the induced IgA by parainfluenza 5 virus against RSV should protect the RSV infection. And PIV5 belongs to the same family to the paramixability to the PIV2, the BCP. The finding encouraged us to develop the intranasal vaccine. And this figure shows the result of a protection in hamster test. That the BCP vaccine strongly protects RSV infection like this, and not in the lung, and also the nasal tubule, as well also in the nasal turbulence at the site of the infection. The protection effect of BCP vaccine failure the potential of the BCP for infants. We are currently performing a GMP manufacturing and the safety testing with a grant from the SCARD and planning to enter phase 1 trial next year. Next, I will talk about the anti-tumor immunization program. I would like to focus on how to efficiently penetrate the TIL into the solid tumor for boosting the anti-tumor immunity. We constructed the BCP with the gene IL-7 and the CCL19 or CCL21. The BCP is intratumorally into the solid tumor. The BCP partially kills the tumor cell and activates the immune cell specific to the tumor. The circulated IL-7 can induce the T cell and the CTL. And the CCL19 or the CCL21 can attract and accumulate the CTL to the tumor surface by CCR receptor on the induced CTL. Consequently, CTL can efficiently infiltrate in the tumor and kill the tumor cell. This figure shows the effect of the animal testing. The BCP were intratumorally administered to the one side of the bilaterally transplanted colon tumor. As you can see, the BCP administration could lead to the regress and bilateral transplanted tumor. Consequently, CTL can efficiently infiltrate into the tumor and systemically and spontaneously spread into the body. The resultant CTL can kill the tumor cell, both treated and non-treated site. Indicating BCP treatment can highlight the induction of the abscopal effect. Addition of the C immuno checkpoint inhibitor will maximize the effect. Red dot of this picture shows many CD8 positive T cells infiltrate around within the tumor, as you can see like this. Next, we were working on the practical application of anti-tumor immune agent. We are targeting the various advanced solid malignants shown here like this. Shown here. And we are currently focused on the breast and prostate cancer, collaborating with the KOL of this field. Currently, we are aiming on the practical application, collaborating with a pharmaceutical company. And this is our management team and advisory. And I am the CEO of this is Dr. Nosaka, Mie University, Dr. Nakajima is Yokohama City University. And last slide, this is RSV vaccine program. We get a grant from the SCADA like this. So now we are continuing to the GMP production. We want to do this program, RSV vaccine program, to the phase 1 clinical. The reason the RSV vaccine for infant is many people want this vaccine. Thank you very much.
Okay, thank you very much, Dr. Fukumura, for the great presentation. I have a quick question about you presented the advantage of the BCP-ID as a nasal delivery. I believe that is the big advantage. On the other hand, do you have any advantage in the manufacturing from the a scale up or other regulatory advantage? Are you facing a difficulty or do you have any challenge?
Yeah, so the parainfluenza virus, there is so far that we have the record for the GMP production. So we collaborate with CDMO in Taiwan. They talk to them. We try to the many, especially the resin. So the reason that we try to the many kind of the resin for the purification for GMP production. So if when we get some record for the specific we have a resin, it is easy to the GMP manipulation. So we try to the many kind of resin for the purification. So and finally we found out that this resin at strong point pivot is separated medium. So after getting the high titer of the BCPF, so after getting the fixed to the purification step, so GMP production is not so difficult we think. So we are try to this one. Yeah.
You're committed to that. Okay.
Yeah.
All right. Sounds good. Thank you very much.
Yeah, thank you for the presentation.
Yeah.
So I have two questions.
Yeah.
So first question is about your next step, your maybe timeline to set up the manufacturer as you just mentioned, and also the clinical trial. Any timeline?
As for maybe for RNA vaccine, or next year we try to the phase 1 clinical. So at first we SCADA. At first we try to the phase 1 clinical in the US or the Australia. But SCADA want to this vaccine try to the Japan. So next year we want to try to the phase 1 clinical trials.
You mean 2026, right?
Yeah, right.
Okay.
So so I talked to the PMDA consultation. They are very interesting about the new type of vector. So,
So second question is about your cancer immunotherapy.
Yeah.
So you mentioned you want to have some partner to form the new corporation.
Yeah.
Can you explain a little bit more about what is the partners you are looking for and what type of biotechs you want to establish?
Also, I still looking for the partner for that. So that reason at the cancer program at the beginning we focus on the developing the vector program and animal model. But we should need last time that we presentation this year we only the animal testing, but it is not so good for the we collaborate with cancer specialist to the focus on the which area. What for example the breast cancer, what area of the breast cancer is needed for the new technology that medical doctor know well about this area. So collaborate with them. We focus on the adjusting our BCP to the cancer. So now we are show you the prostate or the more than the prostate or the breast cancer. Another field we were very, very interested. Some, for example, the lung cancer or the, so we are now we are continue to the collaboration with some KOL.
Thank you.
Thank you very much, Fukumura-san, for a great presentation. I have a kind of confirmation questions. So you said for the cancer, anti-cancer, the vector. So is that intratumor injection or sub-Q injection?
Intratumor injection.
So in the intratumor.
But in case of the sub-Q, we have the cancer vaccine. We try to the many times. That's in this case, sub-Q was intradermal injection. But we have got a good result for the. But CTL is induced. But CTL could not infiltrate in the tumor. The CTL accumulate in the boundary of the cancer. So I show you the IL-7, the CCL19 directly into this one is a CTL actively into the tumor. So we want to mix this technology. So yeah.
Okay, so the back to your inter-tumor injection.
Yeah.
So how do you determine how many shots to one tumor you have to make to get the efficacy? How many places you have to? How do you determine or decide the number of injection to the
so I at the beginning we were not clear about this one. So the KOL, the medical doctor, so the science, so they say that nowadays direct injection technology is a very acute rapidly. So many techniques that it directly into the tumor in the so-called or the tissue. Nowadays it is not so difficult. So far the determine the exact place to injection. It is not so difficult. So
just one injection is good enough to get enough efficacy?
So depend on the animal testing, as only the one injection may be enough. That means the data shows that this is the abscopal efficacy. And this is the T-cell, induced T-cell memory. And after eradication of the tumor, about 1 year later, we put the same tumor cell. There is no regress. That means that this vector, after treated this vector, the T cell memory, I think the strong T cell memory is induced in animal testing. Yeah.
Thank you very much.
One just quick questions. For the cancer vaccine, how big the clinical trial will be? If that will be very, very big, you have to tie up like a big company like Johnson & Johnson.
I want to try to the cancer vaccine. So pancreatic cancer, for example, KRAS is mutation is very critical for the cancer. So now we want to try, but so Biocomo is very very small company. So we want to try, but if possible, many big pharmaceutical company can collaborate with us. We are very happy.
So this is a kind of challenge for any kind of cancer vaccine.
Yeah, that's right. Yeah, so infiltration is important for the CTL induction is so good, but infiltration in the tumor is a problem, we think.
Okay.
Yes,
all right. Then, Fukumura-san, thank you very much for the great presentation.
Thank you very much.
HVC 京都2025スタートアップ起業を志す研究者募集中。 HVC 京都はヘルスケア領域のスタートアップとオープンイノベーションに積極的なパートナー企業やベンチャーキャピタルが国内外から集まるイノベーションプラットフォームです。一流のアドバイザーから海外市場を視野に入れた事業戦略、事業プレゼンのアドバイスなどを受けることができ、事業提携先や共同研究先、出資先を検討しているパートナーとの個別商談を実施しており、多くのマッチング実績があります。詳しくは動画の概要欄のリンクからエントリーシートをご参照ください。エントリーは無料です。創薬、バイオ、医療機器、デジタルヘルスなどヘルスケア領域のスタートアップの皆様、研究成果を実用化したい起業志向の高い研究者の皆様のご応募をお待ちしております。